Our next speaker today is Dr. Finn Hawkins. Dr. Hawkins is an associate professor of medicine in the Department of Medicine and also a physician scientist in the Center for Regenerative Medicine at Boston University, where his lab leverages the stem cell properties of airway basal cells for to develop cell-based gene therapies. Dr. Hawkins' long-term vision is to develop curative strategies for individuals with genetic lung diseases, and today we're going to be hearing him speak about modeling and treating lung diseases with induced pluripotent stem cells. Join me in welcoming Dr. Finn Hawkins.
Hello, everyone. All right, I'm going to change gears here quite significantly. And we've heard about AI and big data, and instead I'm going to ask you to think about one single cell and the journey that it's taken over 15 years. And Chris, I'm going to try to answer your question as well earlier in terms of how good are these cells. So just to really emphasize, induced pluripotent stem cells are really remarkable. This idea that you can take any cell, for example, peripheral mononucleated blood cell, reprogram it by just overexpressing these 4 transcription factors, and that cell now essentially becomes an embryonic stem cell. And why do we like embryonic stem cells? Because in theory, they can make any cell type or any tissue of interest if we can just figure out how to get them to that point. And so our story starts in 2010, when from a blood sample, one single mononucleated cell was reprogrammed into an iPS cell. And we're going to call that cell Jeff. And we're going to follow Jeff over the next 15 years and see how far Jeff came. So my interest has been in making lung epithelial cells from iPS cells. And in particular, we're going to focus on the airways, which are really important in protecting the gas exchanging portions of our lungs. And on this image, you can see what the pseudostratified epithelium looks like. We have ciliated cells, mucus-secreting cells, and this acts as a physical, chemical, and immune barrier to protect our lungs. And all these specialized cell types are actually maintained by my favorite cell, which is the basal cell, which is pictured in this red circle here. These are the stem progenitor cells of the airways. So iPS cells are pluripotent. They can make any cell type in the body. This is a tissue-specific stem cell that can make any cell type in the airways. And it's really a remarkable stem cell. It can heal our airways after all sorts of injuries. And so the goal was to actually make basal cells from iPS cells. And so Jeff, over the course of a decade and through hundreds and hundreds of experiments, was used to develop directed differentiation protocols to make lung epithelial cells, where we gradually were able to make those cells more and more mature, and eventually to make basal cells. And this work is published and exceedingly boring after lunch to go into the details of the direct differentiation protocols. So I'll just prove to you that these cells are actually basal cells. They express all of the key markers that basal cells express. For example, in this image here, the key transcription factor P63. But molecularly, they're highly similar to the basal cells that are in our airways. But the proof is in the pudding. And the pudding for airway stem cells is, can they actually make the tissue of our airways? And this is an image here showing you a mucociliary epithelium. So we're looking at these basal cells that have now been allowed to differentiate in these air-liquid interface cultures, and we're looking down and we're seeing this lawn of cilia. If it was a little darker, you could appreciate, but it looks like the bottom of the seafloor. These are the cilia that beat the mucus, and in the red are the secretory cells. And this looks identical to our human airways. And actually, not only does it form this epithelial sheet, it forms a tissue. And I'm saying tissue because here is a low magnification image of a mucus hurricane. So Jeff has given rise to these basal cells, which have differentiated into an airway epithelium, which is secreting mucus, which has functional multiciliated cells, and the multiciliated cells are actually beating in a coordinated fashion. And here Jeff is on the 17th floor, staring out over the city of Boston 15 years later. And it's quite a remarkable story for one single cell. But we'd like to now apply this to actually, clinical human problems. And so one easy example is cystic fibrosis, where, for example, there are some patients who are very rare, who have mutations that do not benefit from any of the recent advances in therapies, and who it's exceedingly hard to get their cells in order to actually facilitate translational studies for gene therapy, gene editing, mRNA-based approaches. And so what you're actually looking at here is not Jeff. These are cells from one of those individuals with a very rare mutation, where we've taken the cells through our differentiation protocol and generated, again, a mucociliary epithelium, which we can use now for disease modeling. But I would like to go back to Jeff and tackle some really more pertinent questions. And I'm going to tell you about this disease that I've become quite infatuated with over the last couple of years called primary ciliary dyskinesia. It used to be called Carter-Gainer syndrome, and it was initially described in people who might have their organs on the wrong side of their body, and they had these lung problems where their airways would get dilated and be unable to clear mucus, and they get recurrent infection, something we call bronchiectasis. We now know that primary ciliary dyskinesia is actually caused by mutations that affect the multiciliated cells and the function of the cilia, as you can see this beautiful image here. And if you take one of those just individual cilia and you look at how complex it is, it's remarkably complex. So this is the largest macromolecular structure in eukaryotic cells. It's composed of hundreds to thousands of proteins. So it's in our airways, it's in our sperm flagellae, in our ears, and the reproductive systems, but incredibly molecularly complex. And so more than 50 different genes have now been proven to cause this primary ciliary dyskinesia syndrome. And so it's genetically a nightmare. And from a therapeutic perspective, it's very difficult to think of how you might treat this disease. And as there's been this explosion in big data, we're discovering more and more patients and discovering more genes and more variants. And there's this massive problem with figuring out, is a variant actually pathogenic? And I'm taking the example of PCD, but this applies to basically all diseases, right? So we found one such patient, a 32-year-old woman who had a clinical diagnosis of primary ciliary dyskinesia. She has sinusitis, as you can see on her CT scan. She has bronchiectasis in her lungs. She had infertility, and her doc sent a genetic screen, and she was basically unimpressive. The only one thing they found was a variant of uncertain significance in this gene called MCitis. And MCitis is known to be important in ciliogenesis. And ciliogenesis is really complicated, but MCitis is really important just as those stem cells are starting to commit to the ciliated cell lineage. But this individual's mutation was kind of confusing. So it's a missense variant. You can see it here at the very bottom. It's at the very end of this gene. In silico prediction tools, one said it's likely to be benign, and the other said it's highly likely to be pathogenic. And so this represents really one of the big conundrums we have in the field of how do you prove that this variant is definitively pathogenic? And so DJ Wallman in my lab has just been has done a tremendous job with this, and so he tried to address this using our iPSC system. But first, he just asked, well, is there any evidence that this person actually has a ciliopathy, a defect in their cilia? Took their nasal cells, compared to a healthy control, and you can see on the top in green, the control cells make these beautiful cilia, but quite remarkably, her cells had almost no cilia. Amazing to think that somebody might have like virtually no cilia in their entire respiratory epithelium. So that's pretty good data that this person has some sort of ciliary defect. We made iPS cells from them, and compared to healthy controls where you have cilia, her iPS cells didn't make any ciliated cells. This proves that she has a ciliopathy, but it doesn't definitively prove that that one variant is the cause. And so we really wanted to drill down on that. And that's where we went back to Jeff, a totally normal iPSC line that we've validated. We know there's no mutations linked to primary ciliary dyskinesia at all. And we use gene editing to insert the mutation directly into Jeff. And what we found is that the variant is 100% causative for this phenotype, where we see no cilia. We can even confirm this by single-cell RNA sequencing, which not just confirms that there's no cilia, but confirms that that block is extremely early in the program of cells committing to ciliogenesis. And we can do a proof of a preclinical simulation where we can try to rescue this, that both proves that it is indeed this gene that's important, but it's also like a preclinical simulation of gene therapy. So we overexpress the normal sequence of this MCitis gene in our patient's derived cells, and we can see a few cells actually have ciliogenesis rescued. So that was exciting, but it was also humbling because this was very inefficient. And how are we ever going to treat PCD patients where there's 50 plus mutations with a gene therapy? So that's where I'm going to leave Jeff for a little bit now. And we're going to talk actually about potential therapeutic applications of these cells. And this, just to highlight that lung transplantation is a very poor form of organ transplantation. Very limited supply. It's one of the most medically intense procedures that I've ever seen in my life. Survival rates are poor, 50% at 5 years. And of course, the person has to go through many years of end-stage lung disease and high morbidity to even get to that point. Cell-based therapies are really on the rise. Bone marrow transplant is the gold standard, with the first transplant being performed almost 70 years ago, unbelievably, at this point. But we're seeing examples in the field now of many other cell types that are in clinical trials, and we're just on the cusp, I think, of really important breakthroughs for cell-based therapies. So could we actually use a cell-based therapy approach for airway diseases like CF and like PCD, whereby instead of waiting for somebody to have end-stage disease, we transplant in the right cell early enough in the disease course to prevent the development of something like PCD or cystic fibrosis. And this was work that Daryl and I had dreamed up years ago, and that he and Martin Ma really pioneered a couple of years ago. And I'm just going to talk you through that data because I think it's really exciting. So first of all, just like in the bone marrow field, you do need some sort of preconditioning to make space for your cells. And so we're using a detergent here called polydocanol in a mouse model, which does injure the airway epithelium, but that epithelium really starts to recover very quickly. So at 5 hours, we take the first most simple experiment. We're just asking the question now of whether, say, primary airway stem cells could actually be used as a therapy. So we're taking mouse primary cells, labeling them with a green lentivirus, transplanting them at 5 hours, and for the first time really asking, can you regenerate the tracheal epithelium through a cell-based approach? Several weeks later, we dissect out the trachea, and we're going to open it up, and we're going to look for fluorescent cells. And without injury, you can see nothing. And if the room was dark, you'd be stunned by how much green there is up on the screen right now. But we see pretty robust engraftment. This was really surprising. In this case, the majority of the epithelium is actually regenerated by our donor cells. Now, so far, I've just shown you green cells. I can't really justify the use of the word regeneration, but if you take an unbiased approach like single-cell RNA sequencing, you analyze all the cells in the epithelium and you just ask, what cells are there? You have all the expected cell types. And then the most important question is, well, are your transplanted cells different? And the stunning result was that there are no molecular differences between our transplanted cells and our endogenous cells, which is something just incredible to us at the time. Chris, you asked how good are these iPSC-derived cells, and that's something we've struggled with for years because we're relying on in vitro assays and markers, but now we can finally ask if you take mouse iPSC-derived airway cells and you transplant them in, they basically function the same. They give rise to all the expected cell types. They regenerate the stem cell pool, which is really important for durability. And you can even excise the trachea and see, for example, here you'll see there's a light green cell that is beating cilia, so you can actually see that those cells are functioning. And most remarkably, you can look at a mouse 751 days after transplant, and they seem to be perfectly normal and have most of their tracheal epithelium actually has been reconstituted by an iPSC-derived cell, a mouse Jeff. And this mouse submitted no complaints, seemed to be perfectly happy. If we take the 3 stories that I've told you so far, the ability to make stem cells from iPSCs, the ability to gene edit these cells, and not to transplant them, we go back to Jeff, and we actually inserted a PCD-causing mutation, and now we've made these chimeric mouse human airways. So in green here, you can actually see Jeff has given rise to a human multiciliated cell in a mouse trachea, and it's a PCD ciliated cell. And you might wonder, why on earth would you go to all the trouble of doing that? And it's because we want to facilitate therapeutic development for PCD, where there are basically no mouse models and no good animal models. And now more ambitious gene therapy, gene editing, mRNA approaches might actually be able to use this modified chimeric human trachea. And finally, can we actually move towards rescuing disease? So in this last couple of minutes, I'll just tell you about our attempts with Kevin, who's here, Daryl, and Andrew Baracle in my lab, to prove that you can rescue cystic fibrosis in a mouse model before we move to larger animal models. And so cystic fibrosis is caused by mutations in the CFTR gene. And we've got a CFTR knockout mouse here. And the experiment's very simple. We're taking primary mouse basal cells that are wild type for CFTR, transplanting them in. And the difference now is we're asking the key question of, can you actually rescue the function of CFTR? And so here's one trachea that's been robustly regenerated by our donor cells. And the analysis is a little tricky here, but it's important. And I'll just take a second to explain that one of the most important readouts for CFTR function is electrophysiological measure of ion flow, because it's an ion channel. And so in red here, you can see a CF mouse. We're activating the channel with the cyclic AMP cocktail. And you can see the CF mouse, nothing happens. But when we activate CFTR in either the wild type or in the transplanted mouse, you see we get similar current. And that's summarized here. And this basically is a really important piece of data to us. This is the first evidence that a cell-based therapy approach has actually rescued the function of CFTR in an animal model. So we're really excited about this. We think it's a very important, but overall, in terms of our ambition, small step that was supposed to go into the airway, but for some reason it's gone there. I hope I've convinced you that the iPSC field has become a really useful tool that I think as we start to partner with AI, the cells are really coming along and the computational side is really coming along. So I really want to thank Andrew Baracle and DJ in my lab, and of course, Daryl, Martin, and Kevin. And so thank you very much.
Yeah, that was great. What will the first human study look like? If you think ahead to the future, the first time you try to put cells in a human with bronchiectasis or cystic fibrosis, what will that experiment look like?
Yeah, that's a great question that we think about all the time. The reality is that there are many challenges. So likely, these cells won't work that well if you put them in an end-stage lung. They're unlikely able to just completely regenerate the entire lung. And so they're not ideal candidates. So a person with end-stage CF or end-stage primary ciliary dyskinesia is unlikely to be a good candidate, but at the same time, from a clinical trial perspective, the most likely to be approved. So I don't think we know yet. I mean, the first step is animal models, and there's a very good cystic fibrosis ferret model. But I actually think one of the most likely scenarios where you could test this in humans is in the setting of lung transplantation, where you could actually do short-term experiments in somebody who's going to receive a lung transplant anyway. But I don't know. It's a complicated question. Yes.
Just keeping in the theme, I guess, of machine learning and AI, you mentioned the two predictive models that you used had vastly different predictions for your variant of uncertain significance. And I guess, obviously, this deep dive into one variant is labor intensive. If you could narrow down your list, that would be efficient. What kinds of data do you think experimental biologists can collect to help improve these models?
Yeah, that's a really great question. Thank you for setting me up. I think probably, I mean, this is an extremely deep dive into one gene, one patient. It's like the exact opposite of kind of the bigger data approach. But I just think the synergy between being able to leverage AI tools with these reductionist systems, which are labor intensive, low throughput, how to get these systems to be much higher throughput and targeted on the really high priority variants. But if we take the example of PCD, I think that's where it becomes really powerful. We could accelerate many years forward just by being able to do that. For example, the axoneme is really complicated. So being able to use artificial intelligence, machine learning to predict protein interactions to understand what are the most high priority proteins to understand, that'd be something that I think would be really powerful.